Models in CodeOcean make it easy for computational biologists, bioinformaticians, and machine learning engineers to work together to train, validate, and use machine learning models and get full traceability of every part of the process. So here we have our capsule for model selection. This capsule evaluates classifiers on a lung cancer dataset, which we can see here. This capsule is tracked by MLflow, so every run of this capsule is going to create an MLflow run in the MLflow experiment that's associated with this capsule. So how do we tell the system that we want this capsule to have this MLflow capability? So on our environment page, we've included a new section that has a toggle that allows you to then track development and training of models directly in MLflow. So once you toggle this on, with the first run in MLflow experiment is going to be created for this capsule. So now you can develop your code, you can provision whatever resources, either CPU, GPU, whatever you need. You can change this very easily for different runs as needed. We can see in the timeline here on the right-hand side, we already have a successful run with multiple models. And we can also see that 2 of them have already been registered in MLflow. So what does that look like? From any tracked capsule, we can simply click View in MLflow in the header here. And this is going to take us directly into the MLflow tracking UI. So now we are looking at the experiment that corresponds to that same capsule. Here we can see all of our tracked runs and models. We can see that 2 of them have already been registered, the same 2 that we saw in our timeline. The MLflow UI makes it very easy to compare different parameters and metrics. So for example, if we wanted to see all of our metrics here, now we can scroll to the right and see them in our table. Now, if we identify one with the performance that we're looking for, so as an example, we can then go ahead and click Register Model. And when we do this, the registered model gets added to our models dashboard, where we will have the full lineage and traceability of how this model was generated. So let's take a quick look at our models dashboard. This is where you're going to find all of the models in your CodeOcean deployment that you have access to. From here, we can very quickly access information about each model. So let's take a look at one of the registered models from the previous capsule, XGBoost. Here we can access the files, the metadata, the capsule that was used to generate the model. We can see the lineage graph. And of course, we have a link to the model itself in the MLflow UI. We click this, takes us directly into MLflow. We're going to show how we can also track and train models within our Nextflow-based pipelines. CodeOcean pipelines are set up to automatically run on AWS Batch to take advantage of parallel computing. Now we can either use a pipeline to train a model, or we can use a model as part of a pipeline simply by dropping it in, just as we would with data. We can then configure the pipeline for the compute requirements it needs at every step of the process, simply by taking a look at the capsule settings. Now, this particular example uses 2 capsules. So the first generates various JSON files with different hyperparameters, and the second capsule in our pipeline takes those JSON files along with our DNA dataset for cancer classification. Now we can use this simple pipeline to tune the hyperparameters of a model. Just like in a capsule, we can take a look at some of the previous results in our timeline. Every single one of these that you see here, each one of these folders represents a hyperparameter tuning that we've done. Just like before, we noticed that 2 models have been registered in MLflow. We can do this by analyzing metrics in MLflow to see the accuracy of our model. And in the last example I want to show, this is going to be an inference capsule that combines inference of 3 different models, which we see attached to our capsule here. In the data folder, we have 3 different models attached. We also have a TCGA colon cancer dataset as part of our data as well. So here is a script for inference, which takes pre-trained models for colorectal cancer survival prediction on DNA methylation, gene expression, and copy number data. And it outputs the results for us in CSV format. So the result that we've created in the timeline here is for the run directly below it. So let's quickly take a look at this. And we'll also dive into the lineage graph that is generated from this result. Now we can see in this graph how every step of the process from the initial data download all the way to the final result is captured and represented as a node on this graph. So we can hover over any of these nodes to find a link to that asset to take a closer look inside. So now you can very easily run inference and validate your models with others. All you need to do is share this capsule. Quickly look at the share window here. And now your collaborators can easily swap in their own data and click run. So this is an example of what we would call pre-production because you can validate your models on real live data before pushing your model fully into production. If you have any questions about our models feature, please make sure to take a look at our documentation or book a demo with us to get into more detail.